So now we'll start with the genetics. So under genetics, we'll be studying about the basics of genetics, the basic terminologies actually, and then we'll say about the classification of genetic diseases. So under that, we'll be seeing the Mendelian disorders, non-Mendelian disorders, chromosomal disorders, and multifactorial disorders. Then we'll see about the lab diagnosis of these genetic disorders. Then we'll move on to epigenetics and microRNA. So let's start with the basics first. So in a gene, we have a coding region and a non-coding region. And this coding region is the one which is going to be called as the exons. And non-coding regions are called as the introns. So the coding regions actually compose only 1.5 percentage, while the non-coding regions, that is the introns, are going to be composing the rest of the 98.5 percentage. So the maximum amount of the DNA is going to be composed of the non-coding regions. So non-coding regions, we usually have the microRNAs, long non-coding RNAs, transposons. So all these actually help in regulating the gene. Okay, so these introns compose the maximum part. So suppose for the gene A, the dominant allele will be represented by a capital letter, while for a recessive allele, it is going to be represented as a small letter. Okay, so whenever they represent capital, it is dominant, and recessive is small letter. And what is homozygous and heterozygous? Homo, as the name suggests, it is going to be same. So either both the alleles should be of the dominant allele, that is capital AA, and then it could either be that one, or it could also be both the alleles being of the small letter, that is a recessive alleles. So this is the homozygous state. So when both the alleles are of the dominant nature, it is going to express capital A, obviously. And expression is going to be of the dominant allele. And then small a, if both of us is going to be present, again here the expression is going to be of the small a, because nothing else is there, right? But if in a heterozygous state, so heterozygous state is the one in which there is going to be a difference of the different allele combination. So one will be of the capital letter and another one will be of the small letter. So a dominant and a recessive together. So in this case, what will be the expression? So here it will be the dominant allele which will be expressing. So remember this. In a homozygous state, it could be whatever is there, it is going to be expressed. While in a heterozygous state, it is the dominant allele which is being expressed. So this is what we can put it in other words, that a dominant allele is going to express both in a heterozygous state and also in a homozygous state, while a recessive allele is going to express only in a homozygous state. Okay, so this is very important. So dominant allele is expressed both in homozygous and heterozygous state, while a recessive allele will be expressed only in the homozygous state. So next we'll see about codominance. So codominance, in the sense both the alleles are going to be expressed equally. Okay, so this is what we call as codominant, like both of the alleles are going to behave as if they are dominant only, even though the alleles are going to be different. Suppose the best example will be a ABO blood group system. So in ABO blood group, if you see, we have A and B group, AB group, right? So in this, A is of the dominant allele here again, and B is again a dominant allele here. But here, both A and B are being expressed equally and simultaneously to the fullest expression. So this is what we call it as codominance. So the codominance example will be ABO blood group. So next, what is a genotype and a phenotype? So genotype is nothing but the genetic makeup. So genetic makeup is the genotype. So basically, if you are telling AA, so that is the genotype actually. But phenotype is actually the physical expression of the genotype. So whatever the gene is encoding, it is going to be expressed in a physical form. So that is the phenotype. So for example, if you have Turner syndrome. So what is the genotype of Turner syndrome? We all know it is going to be 45XO. So that is the genotype. 45XO will be the genotype, and the phenotype of this 45XO will be the morphological features we see in the Turners, like low hairline, then they'll have the webbing of the neck. So these things and streak ovaries, all of these are going to be the phenotype. Okay. Next, we'll see what is Lyon's hypothesis. Lyon's hypothesis states that even though there is a XX chromosome, XX expression, one of the X is going to be inactive. Okay, so in the X, it is going to be represented as XX, right? So the X gene is going to be expressed as XX alleles. So out of this, one of the X is going to be inactive, and that inactivation of X, whichever X, is going to happen at a random. Okay, so random inactivation of the X usually happens on day 5.5 of the embryo. So this is again an MCQ. So random inactivation of the X is going to happen on the day 5.5 of the formation of the embryo. And this inactivation is actually caused by silencing. So silencing of this X is going to happen by methylation. So methylation, we already know, methylation is a epigenetic change. Here this methylation, whenever there is methylation, DNA methylation, it is going to result in the silencing of the gene. So here the X is going to be silenced, causing the inactivation. So this X inactivation is going to be controlled by a long non-coding RNA called as ZIST. Okay, so XIST is ZIST. So ZIST is going to control this random inactivation of the X. And remember, this ZIST is going to be a long non-coding RNA. So link RNA we call it. It is actually the short form for long non-coding RNA. So the number of the bar bodies is going to be usually number of the X minus 1. The formula for calculating the number of bar bodies is going to be X minus 1. So bar bodies are nothing but this inactivated X only. So whatever X was inactivated, it is going to form a bar body. So what is this bar body? It is going to be present. You can visualize this bar body in a peripheral smear. So in the neutrophils, if you see in the nuclear lobes of the neutrophil, attached to the nuclear membrane, you can see a small structure which is going to appear like a drumstick. So drumstick appearance of the bar body is going to be seen in the neutrophils nuclear lobes attached to the nuclear membrane. Okay, so this is the classical description of a bar body. So the number of the bar bodies you can count by calculating the number of actual X minus 1. So a female, normal phenotype of a female will be a 46XX. So how many bar bodies will be present in a female? So we have 2X. So 2X minus 1 will be 1. So in the normal female, you are going to have 1 bar body. In 46XY, that is a normal male. So in a normal male, you are going to have X minus, here only one X is there. So minus 1 will be 0 bar bodies. And in Turner syndrome, we know it is going to be represented as 45X. So here again, there is going to be only 1 X. So number of bar bodies will be here again 0. Okay, understood the concept of bar bodies now. So next we'll move on to the types of chromosome. So based on the position of the centromere, we have 4 types of chromosomes. So we have metacentric, submetacentric, acrocentric and telocentric. So metacentric is the one here wherein the centromere is going to be present at the center, more or less at the center here. So the example of this chromosome will be chromosome 1. Okay. Submetacentric, as the name suggests, it is going to be near to the central position. So if this was the center, somewhere near to it, you are going to have the centromere. So that is submetacentric. Example is going to be chromosome X. Acrocentric is one in which the centromere is near a particular tip. Okay. So this was the center. So it has moved way upwards and it is more or less near the tip. Okay. So that is acrocentric. So many chromosomes are of acrocentric group. So we have chromosome Y, chromosome 21, 14, 15, 22. So all of these are acrocentric chromosomes. Telocentric is the chromosome in which this was the center. Here the centromere is exactly at the tip. Okay, so one of the ends is going to form the centromere. So tip at the tip is the telocentric. Here, this is not going to be seen in humans. So telocentric chromosomes are not seen in humans. So next we'll see about karyotyping. So what is karyotyping? So karyotyping is nothing but the study of chromosomes. So to study this, we are going to take samples. So what kind of sample we can take for karyotyping? We can either do a chorionic villus biopsy or amniocentesis. So when you are going to detect some diagnosis, prenatal diagnosis, suppose the parents are recessive, so both the parents are recessive, they want to check whether their kid is going to have the disease or not, they'll go for prenatal screening. So in that prenatal screening, you'll have to check for the chromosome. So for that, you are going to do this chorionic villus biopsy or amniocentesis. And then you can also use the peripheral blood lymphocytes. So in the blood, you can use the lymphocytes as well, but remember, you cannot use the monocytes. It's the lymphocytes which are being used. Other than that, we can also use the shed skin fibroblasts as well. So this was also asked as MCQ before. So all this for as a exception type of questions, these options were given. So usually we view the karyotype, that is the chromosome, we view it at the metaphase. So at the metaphase, you are going to visualize the chromosomes. So you are going to stop the mitosis at the metaphase by using the colchicine. So arrest is done by a colchicine. Okay. So for after this, we'll have to fix the chromosome. So the fixative used here will be the Carnoy's fixative. So Carnoy's fixative, it has methanol and glacial acetic acid. So methanol to glacial acetic acid will be in the ratio of 3:1. So all of this again has been asked as MCQs. So for visualizing, you will have to stain the chromosomes. So the most common staining method is with the Giemsa stain. So with Giemsa stain, it is a Romanowski stain, right? So with Giemsa stain, the chromosomes are going to appear like bands. So band of dark band, light band. So this is why it is going to be called as G banding. So G banding is the most common method in which we view the chromosomes. So G banding is nothing but Giemsa banding. So we can also have other stains for visualizing the chromosomes, which is Q banding for quinacridine, R banding for reverse banding, and C banding for centromere banding. So quinacridine is going to be a fluorescent dye. So you have to visualize it under the fluorescent microscope. So Q banding, R banding, C banding, and the most common one is the G banding, which is for Giemsa. So let's see about the examples of karyotype. So you can get image-based questions from karyotype. Okay. So in this image, if you see, the chromosomes are going to be arranged under descending order of the length. So the chromosome 1 will be the longest chromosome, while the chromosome 22 is the smallest chromosome. So with the length arrangement, it is going to be arranged. And here X and Y are the last chromosomes. In this case, if you see, all the chromosomes are having all the chromosomes are having two chromosomes, right? At the 22 sites are there, so everything is having 2 chromosomes. But in X, we are going to have 2X here, and Y is 0. So what is this? So here it is going to be 46XX. So what kind of phenotype is this? Genotype is this? This is a normal female. Okay, so this is the karyotype of the normal female, 46XX. So in the next image, if you see, you are having 3 chromosomes here at the chromosome 21. So what is this? We have trisomy 21, which is nothing but Down's syndrome, right? Down syndrome is trisomy 21. The next image, if you see, you are having the 3 chromosomes at 18th site. So here it is going to be trisomy 18. So trisomy 18 is called as Edwards syndrome. The next one, if you see, here there are 22 chromosomes are going to be normal, but the X chromosome, there is only one X chromosome, there is no Y chromosome. So this is 45XO. So only one X is there. So XO. So this is Turner's syndrome. The next image, if you see, there is 3 chromosomes at 13 sites. So here it is trisomy 13. So trisomy 13 is Pathology syndrome. Okay. So next one, if you see here, you are having all the autosomal chromosomes which are normal. But at the X, you are having 2X, and again in Y, you are having 1Y. So it has become XXY. So it is going to be 47xxy. Okay, 46 is already because of this entire thing, and there is an extra x here, so it is going to be 47xxy and not 46xxy. So this again students tend to make mistake. So 47xxy is the genotype for Klinefelter syndrome. So Klinefelter syndrome, you are going to have an extra X. So normally a male is going to have XY, and Klinefelters you can have either 1 or 2 extra X. So it can either be a 47XXY or 48XXXY. So triple XY, then the case you will have to increase one other number. So normal male is 46XY, Klinefelter is 47XXY or 48 triple XY. Okay. So now we have seen about karyotype. We'll see what are mutations. So mutations are nothing but abnormal changes which happen in a DNA, and these changes are going to be permanent. So we are classifying mutations into point mutations and frame shift mutations. So point mutation is going to be the one in which the reading frame is not going to be altered, but a position of a single nucleotide is going to be altered. So it could either be a substitution. So if suppose I'm replacing one of the nucleotides, so I suppose I'm replacing a thymidine with the adenine. So in that case, T is replaced by A. So the sequence of the protein is going to be, the amino acid sequence is going to change. So some other protein is going to replace it. So when amino acid sequence changes, the function of the protein which is being formed will also be changing. So the result of this point mutation is usually either a silent mutation or a missense mutation or a nonsense mutation. Silent mutation is the one in which, even though there has been a substitution, the change in the amino acid sequence has not resulted in a overtly apparent phenotypic change in the individual. So that is what we call it as a silent mutation. There is going to be no change. While a missense mutation is one in which the substitution of one of the nucleotides for some other nucleotide is going to change the amino acid sequence now, and it is going to cause a different protein to be formed altogether. So the different function is going to be formed here. So the best example of this will be in sickle cell anemia. So in sickle cell anemia, we all know it is the beta 6 position. So sixth chain of the beta chain of hemoglobin. Here at this position, it is the normal chain had glutamate, but here in sickle cell anemia, glutamate has been replaced by valine. Okay. So valine replaces glutamate at the sixth chain of the sixth position of the beta chain of hemoglobin in sickle cell anemia. So when this valine has replaced glutamate, it has resulted in the formation of HBS. Okay, so HbA has become HBS. Hemoglobin A has become hemoglobin S. So it is a significant change. So because of this HBS, there is going to be lot of problems for the patient. So sickling is going to happen, and as such, the properties of HbA and HbS are completely different. So this is an example of a missense mutation. So coming to nonsense mutation, what happens is, with the substitution of a nucleotide, there is going to be formation of a stop codon. So stop codons are usually UGA sequence or UAA sequence or UAG sequence. Okay. So these three sequence, if any of the nucleotide is inserted, changed. So if that results in the formation of this stop codon, it results in the premature stopping of the protein. So a truncated protein is formed. So it doesn't have any meaning. So it doesn't form any proper protein. So this is going to form a nonsense mutation. So there is no sense to this. So the best example of this is going to be thalassemia. So thalassemia is an example of nonsense mutation, while sickle cell anemia is an example of missense mutation. So frame shift mutation is the one in which there is going to be an insertion or a deletion. Okay. So normally we know that for every 3 codons, it is going to, the sequence is going to be read. So for every 3 codons, it is going to be read as an amino acid protein. So this amino acid sequence is read like that. Suppose I am going to insert one other nucleotide here. So suppose CAA. So this here the reading frame changes to CAA rather than CAG. So CAA is going to encode to some other protein. Okay. So the entire reading frame had changed. Even if in deletion, suppose CAG AT, suppose it is like this, and I am going to delete this G. So what happens again here, the reading frame which was here is going to be read as CAA. Okay, so the reading frame is going to alter, which will result in the difference in the reading of the amino acid sequence as well. So that is going to result in lots of problem again. So that is called as frame shift mutation. Here it is due to insertion or deletion, while point mutations are usually due to substitutions. So now we had seen. So one other thing I wanted to mention is about pleiotropy. So pleiotropy, in the sense, a single gene mutation can result in various manifestations of the disease. Okay, so suppose for example, if you take a Marfan syndrome. So Marfan syndrome is an autosomal dominant disorder. So here one of the patients is going to have a tall habitus. So the patient is going to appear very tall. So Marfanoid habitus. And some of the patients are going to have cardiac defects. So the same gene mutations, it is the fibrillin gene mutation only, but having various defects in various, even though the disease is the same, various manifestations are going to be present. Okay. So same gene mutation having various effects or manifestations. So this is what we call it as pleiotropy. Then we have something called as heteroplasmy. So heteroplasmy is actually a property of mitochondrial disorders. So in mitochondrial disorders, it is usually the mitochondrial DNA is going to be damaged. It is the defect in the mitochondrial DNA. So normally in the mitochondrial DNA, you are going to have both normal and the mutant mitochondrial DNA at variable proportions in the same individual. Okay. So you are going to have both normal mitochondrial DNA and abnormal mitochondrial DNA present in variable proportions in an individual, resulting in mitochondrial diseases. So because of the difference in the proportion of this normal to abnormal mitochondrial DNA, the manifestations of this mitochondrial diseases is again going to be different in different patients. So it is same as pleiotropy only, but we use the term heteroplasmy in mitochondrial diseases.